closer look at species analysis for plants or floral remains. This is when our lesson gets a little bit more into vocabulary that we don't normally use very often for things. So I want to be sure that you're clear and that it syncs with your reads. All right, so we are going to go over this a little bit quickly for some because we did look at this previously, but then we'll slow down when there's something new. Floral analysis, also archaeobotany. What were ancient populations doing with plants? So we took a look at the question of the importance of carbonized samples. And so when things are heated, burned, and then cleaned up or left behind in fire pits, midden, and so forth, then we do end up with things that tend to preserve fairly well. We also talked about one of the key things before we do any other further investigation on either floral or faunal analysis is determining the species that we, and particularly understanding that there are differences between wild versions of a species versus that period of time when people begin to accidentally genetically modify their favorites, leading to a end result, which we call domesticated versions. So, difference between a wild goat and a domestic goat, or a wild dog and a domestic dog, or wild seeds versus domesticated corn plants. Also, one of the things right off the bat, once we see the species, then we have a stronger line of information about seasonality. When are these plants or animals in use? And does that influence where they're living, where they're traveling, and how they are planning to use their time during the world? During your year, are there times where they're going down to the ocean, higher altitude, leaving their zone altogether and coming back later, sending work teams out? All right, next one, still on floral remains. Seasonality, and you do have this from previous, when that particular species is available and where they're going to get it. So if it's something that's not 12 months per year present on the landscape, then we've got a lot more information about movement, work assignments, ranked decisions about what they're going to do at what time. And then as you go through your reading, they have some information about how those eco-facts, sometimes they're artifact data, but a lot of times eco-facts are collected. And we looked over quickly flotation, collecting soil or other types of softer samples, putting them in water, adding something to the water that causes the organics or the lighter, very lightweight things to float up to the top so that they can be scraped off, dried, and then looked at under the microscope. So this is the part where there was a question, phytoliths. So let's compare, contrast, or at least highlight phytoliths versus diatoms. And then there's another one. So let's, because those are in your reading. Okay, so we did look at this image, phytoliths. Phytoliths are inclusions, silicate inclusions that can be found in some, as you can tell, super microscopic plant remains. Finding them might help us get a date or may help us identify. Okay, so phytoliths, microscopic, right? So these are going to be micro data, pieces of silica. Silica is sand-like. that will be formed in the cells of many plants. What do we use phytoliths for? We use phytoliths to identify the species of plants. They will frequently give us information on shifts in vegetation or climate over time. And we can collect them even from the sediments that slowly deposit, or sometimes in larger quantities in completely waterlogged environments such as lakes. If it's a river, right, rivers have a push, and if they're larger or flow faster at certain times per year, those things tend to get pushed far from their original source. But in lakes and ponds, things come in from after it rains or in the wind or just settling down in the air, get down to the bottom and form part of the strata. So that gives us date. When we have strata, we have time. Okay, phytoliths. So they're like pieces of hard glass. They remain very well preserved for very long periods of time because they were an internal part of the formation of the plant parts themselves, like leaves, stems, stalks, fruits, and so forth. So the plants die and decay, but these phytoliths can be left behind. So real quickly here, what would you want to have here? Most common uses. So why do we care? Why are we looking at this in class? Firstly, identification of, and this is a science term for you, taxa. Taxa, those of you who have had a Gen Bio course, taxonomy is the science of classification of living things. So when we know what groups they're in, like the species or the genus or the family or the order, then our ability to have much better analysis moves forward. Now, once we identify the plant taxa, what type it is, then we might be looking at what humans ate, farming practices. In this one, typo, sorry about that. Farming, foddering. So just in case, fodder, when they are collecting or raising plants, not for humans to eat, but for their animals. So fodder, like if they're feeding sheep, goats, cattle, horses, camels, donkeys. Yep, so that's fodder. Or, and also very importantly, want to highlight that we get better information about construction materials. So if we see a lot of it, we know it's not something that's part of the diet, then it begins to give us a better avenue to understanding construction. Where did they get materials? What kind of materials? What else was needed? Secondly, when we can collect and identify phytoliths, then we may also be able to be more fine scale analysis to know what part of the plant they were collecting. So a good example would be, and in our unit 2, as we move towards looking at the Neolithic in the Middle East for the beginnings of raising your own food and storing those foods for months, either to consume later in the year when there's less natural food or to use in farming season the next year. So for instance, if we're getting plant parts from the large leaves that come over, let's say for instance, corn cobs in the Americas, or around the little leaves around wheat, barley, oats, and so forth. They don't eat those parts, they're gonna remove them, but those are going to tell us the species and what they were doing in that building or in that jar. All right, that was hopefully a little more clear on phytoliths. Next one. We did look at that phytoliths are an important modern type of specialty for the floral analysis specialists for archaeology, and that there's even a modern book, an archaeobotanist's best friend. So again, we want to know what they ate, but we also want to know when they might have started with interests in certain food sources or sources for their animals, if they are keeping animals. When did they stop moving around so much? Or are they still moving around the time frame we're looking? And again, what are phytoliths? What are those telltale clues? Now, in your reading, they do also mention two different things, and I saw a question there from text. And so there's a couple of other words, but again, our foram, and this one, I need to go down. Typing. Typing when you're trying to go really fast. You think you see the right spelling, and then later you look at it again and say, yeah, needs an upgrade. Okay, foraminifera versus diatoms. These are also microscopic ways to take a look at things. As we were looking at phytoliths in general, how might these be used slightly differently in your reading and really efficiently for you all? What might you want or need to do with these for your notes? That phrase, plant taxa, right? Species, genus, families, orders, suborders, those sorts of terms, right? Foraminifera are single-celled organisms. So they are related to the earliest forms of life on Earth, and they are very tiny. So you have to see them again. These are microfossils. So when you see this in reading, they're just identifying that we can determine certain single-celled species of organisms. And in this particular case, they are they get this particular term because when they look at them under the microscope with enough magnification, they can see these sort of little streaming particles. But just like pollen, just like silica, just like diatoms, different species create slightly different shapes, sizes, or surface distinctions to them, so they can be categorized or identified. And that gives us information about, for instance, where they come from. So in foraminifera, these are going to be organisms that live in water. And so when we find their remains in things, we know that they were originally brought in from water. Or the area that we're excavating used to be standing water, shallow or deep waterlogged. Now, diatoms are just the remains of organisms that we now call algae or algae, depends on what continent you're on. And for you all, so they are just, so the only thing that you really need to know, these terms, foraminifera, diatoms, phytoliths, all about determining plant species with microscopic data. So diatoms are just different species, and both are used to determine species, right? So species identification and what reasons they might have appeared. So we're just getting more data, more plant-related data. So hopefully that answers the question that I saw last time, and answers another question. How much detail do I need to know with this as I finish making some notes from chapter 6 and move on to the next part of what we're going to take a look at, which is cognitive